I had no idea. I didn't see it. I just thought it was. I didn't assume that that's fake. It's not a short too.
Oh, yeah.
All right. Skeleton crew that we have. 45.
No, James.
James not allowed. They apparently got up and beat the break. Kylie, I saw Kylie yesterday. She was pale, peaked, and she wanted to do the lab. So I had her do the lab, and she did not look well. Are you feeling better? She said, not really. Apparently, the whole class. Do you want to practice, say the first hour and one slide? Again, Emma, can you hear me?
Yes.
Yes, we can.
Congratulations on your wins.
Thank you.
We're pulling for you on Saturday, of course. And then, of course, on Monday after you win, right? So I said I'd do a Blackboard quiz. I guess I lied. So I will email you guys the quizzes and then have you guys just take it and return it to me. And this will be on your honor. All right.
Okay, perfect. Thank you.
Right. Yeah. Don't. Make sure you flip it over. That's your over at the bottom.
Yeah, you guys have more questions.
We didn't hold this time. All right. Gonna finish 17, get into 18. I need to mention that next week you have a group project that's due. That group project is a brochure that you need to make as a class. The brochure is meant to advertise essentially to a young family why they should get their child vaccinated. And the scenario is in the syllabus. How you do the layout, what information you put in there. There's lots of great public information available. CDC has all kinds of good information on vaccination. So does the American Association of Pediatrics. And there's some other sites as well. Again, you have a mother who's waffling on whether or not to vaccinate her kids. You want to hand her something that she can easily read that would be convincing. Artwork is great, lots of color and lots of things that are eye-catching. Try to make it objective while making it eye-catching and not over the top. You're so dumb if you don't want to vaccinate your kids. No, that's not going to fly. Something, and so some of you are probably very artsy. Some of you layout and that sort of thing is something that just kind of comes second nature to you. Others of you are not quite as artsy or more, I can do the writing. Others of you are more the managerial type. You are probably better at keeping the whole project on track. So that's due end of next week. If you need more time, we can make that due the following, because the last day of classes would be the following Monday. So if you want it to be due, say, before finals, that's fine if you need more time as well. So I haven't said much about that since I last talked about it when we went over the syllabus, but that is a project you guys will need to have done by next week. Lab, if you got my email about lab, so lab is pretty much done. What remains to be done is cleanup. If you have any cultures that need to be pitched, make sure you go in and pitch them. If there are any messes that you made, please clean them up. If there's a big stain on the bench and you're pretty sure you made it, or even if you're not sure, you're not so sure you made it, it's still on your bench, so you're probably responsible for it. Please come in, wipe your bench down. If you need some alcohol to get stains off, that's fine. Check your drawer, make sure your drawer has all the equipment. If you have duplicates of equipment, give them to me and I will store them or give them to someone who doesn't have something. If you're lacking something, let me know. I'll make sure your drawer is good for the next semester. And that is something that is your responsibility, is your drawer. And then if you made a mess in the refrigerator because a culture spilled or something like that, please clean that up as well. Just use cleaner, clean it up. So the old dictum, clean up your messes, your mother doesn't work here, applies. And that's pretty much it. And if your stuff's all cleaned up, really, you don't need to come in next week. Unless you really, really, really want to come in which case, yes, I'll be there. But you can clean up another time, so a little extra sleep might not hurt. Yes, well, one can hope. Hope springs eternal. All right, let's review very briefly our meningitis infections. So from 0 to 3 months, the main culprits will be strep group B, streptococci, that's Streptococcus agalactiae. You saw that organism in the lab. Listeria monocytogenes, if the baby gets it from the mother. Remember, that's one that can cross the placenta. From where do you get listeria? Sorry? Dairy product, contaminated dairy product. So food, you get it from food. Contaminated dairy, deli meats, soft cheeses, that sort of thing. And if the mother gets infected, she may not know she's infected. She may have a subclinical diarrhea, but it can cause then a very mild bacteremia that doesn't really cause any symptoms. It can cross the placenta, cause a very, very severe neonatal meningitis. From 3 months to 12 years, now the more common culprits will be Streptococcus pneumoniae, Haemophilus influenzae, unless the child has been vaccinated, and Neisseria meningitidis. That would be 3 months to 12 years. Then 13 to 17 years will pretty much be the same as adults. It'll be Streptococcus pneumoniae, Neisseria meningitidis, Haemophilus influenzae. If someone has had any trauma to the head, penetrating trauma to the head, the main culprit will be Staphylococcus aureus. And if they have, say, been camping, say, to the northeast of the United States, and they have a tick bite, they get Lyme disease. Lyme disease can cause a meningitis. Also, if they've had a untreated syphilis condition, that can cause a meningoencephalitis as well. Or if they've been to Central or South America and they were bitten by a dog tick, they can get Rocky Mountain spotted fever. Rocky Mountain spotted fever can also cause a meningitis. With Rocky Mountain spotted fever, you will usually have a rash that starts at the ankles and wrists and then extends usually to the body. It goes from there to the body. And that is spread by the dog tick, dermacenter. If you culture the CSF and you don't get any organisms, it is called an aseptic meningitis. What will cause an aseptic meningitis? Just think of the things that don't grow in petri dishes. Fungi. Typically, fungi will grow and then people will throw them away. Oh, it's just a contaminating mold. So it's not really an aseptic meningitis, but what? Okay, fine. It's not bacterial, so fine. It's an aseptic meningitis. What's the biggest cause of fungal meningitis? It's a yeast in the body associated with pigeon dung. Starts with a C. Not Candida, no, not Candida. Candida can cause meningitis, particularly in immunosuppressed people. Cryptococcus neoformans, Typically starts with pneumonia. Very often that pneumonia is completely subclinical, although in some people it can be a robust pneumonia. The organism has a predilection for the brain. You get hematogenous spread to the brain, particularly an issue in people with HIV/AIDS, solid organ transfer patients, cancer patients, and people on immunosuppressive drugs or who have inherited immunodeficiencies. Then the viruses, it's usually going to be the enteroviruses. So echoviruses, par-echoviruses, Coxsackie viruses in kids, HSV, and varicella zoster virus can also cause encephalitis in kids. If the kids are older, it's usually self-limiting. If the kids are really young, it can be severe. Remember, HSV and ZVZ, those can be passed from mother to infant. In adults, arboviruses that are passed by mosquitoes The big one in adults, if elderly adults, will be West Nile. West Nile usually in most people causes a completely asymptomatic infection. You don't even know you have it. And your immune system usually deals with it and it's fine. In elderly people, it can cause encephalitis. West Nile virus encephalitis will always cause tremors. That's one of the features of West Nile virus encephalitis you can count on is the tremors. All right, so we talked about a lot of this already. Talked about these infections here, Cryptococcus. Coccidioides we'll talk more about when we get to pulmonary infections, because this is a pulmonary infection in immunosuppressed people and in people of certain ethnic groups. It can cause meningitis at higher levels. Talked about Echolactia, E. coli, Chronobacter. Where do you get Chronobacter from? It's freeze-dried. You feed it to babies. Formula. Contaminated baby formula. Chronobacter sakazakii. Zika virus. You get this from mosquitoes. This is another flavivirus. Mothers don't know they're infected, but it can cross the placenta, infect the baby, and cause microcephaly. So in areas where Zika virus is endemic, the mom should wear long sleeves. She should also wear insect repellent. Talked about polio. Talked about primary amoebocytic meningoencephalitis. All right, here we go. So as we said, pure encephalitises are almost always caused by viruses. Usually in healthy people, encephalitises that are severe are rare. If you see severe encephalitis, it's usually in someone very, very young, infant, say to 3 months, or on the elderly end of the spectrum. So in elderly people, West Nile is one of your biggest ones. Since West Nile is passed by mosquitoes, what can you do usually to limit your exposure to mosquitoes? Sorry? Repellent, insect repellent. So one of your jobs as a nurse is to remind people who are in higher risk groups, and if you do family medicine or something like that, having talks with people about insect repellents and which ones to use. Consumer Reports has several good reports on different types of insect repellents. The one that is the best in terms of efficacy is DEET. DEET has kind of an offensive smell to some people. Some people also, it irritates their skin, but DEET is safe if you use it as directed. If you put too much of it on and it soaks in, it can cause seizures in some people, but if you use it as directed, it comes in lotions or sprays or something like that. You can spray your clothes with it. It is safe. So if used as directed, it's safe. Citronella is also safe, but some people develop allergies to it. Citronella also is not as effective as DEET. That's pretty clear. JC virus stands literally for the John Cunningham virus. The John Cunningham virus. So this is a double-stranded DNA virus. And there is a closely related virus like JC virus called BK virus. So my students one year jokingly called this the Burger King virus. And if that helps you remember it, fine. Go with it. These are viruses that almost everybody has. So JC virus, for example, infects the kidney. And then your CD8 positive cells, those are your cytotoxic T cells, destroy the cells that are infected with it, hold it with bay. Then it becomes latent in the kidney. Virtually all of you in this room have it. If you become immunosuppressed, now this virus can come out. It can cross the blood-brain barrier and it can infect the brain and cause, in some cases, a very serious encephalitis. If it infects the brain, it will attack oligodendrocytes that myelinate cells, and it can cause a potentially very severe condition called progressive multifocal leukoencephalopathy, which is mercifully abbreviated PML. So that's progressive multifocal encephalopathy. PML. This is particularly the case in people with multiple sclerosis, because, as we'll find out next semester in Pharm, the most effective drugs for multiple sclerosis prevent immune cells from accessing the central nervous system. Without those immune cells in the central nervous system, you can't deal with JC virus in the central nervous system. So PML is a potential side effect of these anti-MS drugs. But it's also a side effect of people whose immune systems are suppressed for medical purposes or because of genetics or something like that. BK virus can do the same thing. BK virus is also a virus almost all of you have, because you've all eaten at Burger King. No, not really. These are small double-stranded DNA viruses that virtually everyone has. So the symptoms of encephalitis will look a little bit like the symptoms of meningitis, except you do not get nuchal stiffness. And you also get behavior changes and confusion because of inflammation in the brain parenchyma. Now, in the cases of encephalitis because of herpes viruses, intravenous acyclovir is effective. I have to say that acyclovir is a drug that, if it builds up in the kidneys, it can precipitate in the kidneys and it can cause renal problems. So if someone has renal problems with this drug, you need to really adjust the drug down. Because if it builds up in the kidneys, you'll get precipitates in the kidneys. They can be very, very damaging to the kidneys. So be careful with that. Doesn't happen with oral acyclovir. It happens with high dose intravenous acyclovir. So just put a little note next to this with acyclovir. Be careful of kidneys. If your PML is due to JC virus and it's due to a medication. Withdrawing the offending medication is really the only way to treat it. Most treatment is supportive. Cyclovir usually won't harm people infected with other agents, but it won't help them unless they have bad kidneys. If you have a flavivirus that is due to a virus that was given you by a mosquito bite, usually the impact on humans is subclinical, but in some cases that impact can be greater if the patient is older. You usually have an acute fever, often accompanied by a transient rash. The rash is usually a blanching rash. That is, you put your hand on it and take your hand off, you see an imprint of the hand, and then the redness returns. These rashes are very nonspecific. And it's usually a mosquito bite. You get some sort of viremia. So you have prodromal types of symptoms that are very nonspecific, usually very flu-like. And it's young children and the elderly who are most sensitive to injury. Most people will show no symptoms. It's very possible a lot of you in this room have had infections with these viruses and showed absolutely no symptoms. Symptoms, as you can see up here, are quite variable because it depends essentially how robust encephalitis is. Best control measures is don't get bitten in the first place by using bug spray and things like that. HSV encephalitis is more common in children, and it's more common with HSV2. Since HSV2 is more likely to cause genital herpes, children usually acquire it either by passage through the birth canal, or it will pass the placenta in some cases. Older adults, if they have HSV latent, usually in the trigeminal ganglion, if they become immunosuppressed, they can suffer from herpes encephalitis. Varicella zoster can do a similar thing as well. That's rare, but it can occur. So JC virus, as we said before, this can cause PML. Here's better. These are just some electron micrographs of the virus. You can see it's a really small virus, small genome too. It's uncommon, but if PML develops, it's usually fatal. In people with HIV, there is a treatment called IRIS, I-R-I-S, that stands for Immune Reconstitution Inflammatory Syndrome. And people who are recovering essentially from a knockdown of their immune system can produce a very intense immune response against the JC virus. Most of the drugs for this that have been used are experimental. So Cidofovir and Cidofovir are two drugs, but they're experimental. They don't have any demonstrated efficacy. Subacute encephalitis, this is usually with toxoplasma. You can also get encephalitis, as we said, with persistent measles infections. Prions. Prions cause Creutzfeldt-Jakob disease, spongiform encephalopathy. That takes usually years to develop. You usually get it through ingestion of the prion protein. It's able to cross into the bloodstream and then into the bloodstream barrier. It will cause normally folded proteins to then start to assume this altered conformation that causes them to have this conformation that now fall out in solution and cause the death of neurons. So toxoplasmosis in healthy people, you can get this, you acquire this usually from cats. It infects a variety of animals. People usually get it through exposure to cat feces. Pregnant women, therefore, are usually told they shouldn't be changing the litter box. If you do change the litter box, to wear gloves or even a mask, because the cysts can be inhaled as well and cause a upper respiratory infection. This is a metazoan protozoan. You can see it here stained with a right stain. They're kind of shaped like sausages. These will tend to cause brain abscesses. Now, if you look at these with an MRI, you see the kind of halo look to these abscesses. That halo look is diagnostic for toxoplasmosis, toxoplasma abscesses in the brain. Toxoplasma abscesses. So your abscesses, in cases like this, you have infection of the brain. The immune system, however, walls it off. But now you have this mass in the brain that's impinging on portions of the brain. It does not tend to increase cerebrospinal fluid pressure unless it's really big. But as it says in your book here, people when they get their toxoplasma infection are more likely to display thrill-seeking behaviors and slower reaction times. That's due to interference with neurological activity in the brain due to these abscesses. So kids who are the victim of toxoplasma infection through the placenta, there's usually retinal damage, blindness, and I told you about my experience. I'm blind in my right eye due to toxoplasmosis probably. Then you can have hydrocephaly, convulsions, liver spleen enlargement, stillbirth, so on and so forth. Pregnant women have about a 30% some odd chance transmitting the infection to the baby. And again, infections in the mom are usually asymptomatic, or low-grade fever, some sort of upper respiratory something. They may dismiss it as a cold. So the cat will usually get it from feeding on pigeons or mice or something like that. So I grew up on a farm. We had barn cats that fed on rodents and birds and stuff like that, and other things, grasshoppers in the garden and stuff like that. So these animals are infected with the organism. These infect the cat. The cat will shed the organism from their fecal material in the form of these cysts. The cysts can be picked up by other animals, or people can get it. It's pretty much a dead-end host. It's not like this infection cycle that you get in animals. In the dead-end host, essentially, the immune system usually handles it, but women who are pregnant can pass it to their baby.
And would indoor cats have a chance of having that then?
If they venture outside, yes. If they don't venture outside, unless they feast on mice inside the house, typically not. All the same, usually as a medical professional, your motto will be better safe than sorry. So you will still tell expectant mothers to make sure they don't change the litter box. And if they live at home alone or something like that, then you will ask them to don protective gear, wear rubber kitchen gloves, and wear a mask if they do change the litter box. Or if they really feel adventurous, they can hire someone to change the litter box, which I've seen that done too. So. All right, so humans, it turns out, because of our association with animals, we get constantly exposed to toxoplasma. Hygiene is a good way to avoid it. Toxoplasma, fortunately, is very treatable. It's very susceptible to sulfa drugs, and that is the mainstay treatment for toxoplasma. We talked about SSPE when we talked about measles. This again is a sequela that occurs years after the original measles infection. Prions, we went ahead and talked about. These cause Creutzfeldt-Jakob disease. So these essentially will cause these plaques to occur in the brain. These will kill off nearby neurons. Will also cause the formation of neurofibrillary tangles. You get these from contaminated meat, it seems. There is an inherited form of this condition where the precursor protein, the amyloid precursor protein, has mutations in it that cause it to spontaneously fold. The dietary link between contaminated meat and human prions, your book says it's well established. I don't agree with that. I think it's not terribly well established, but Stanley Prusiner won a Nobel Prize saying it was well established. And the force of his personality tended to drive the field to that conclusion. If it is linked to animal meat, it's not. It probably takes years and years and years and years of eating animal meat that's contaminated with this to get this. So eating one hamburger is not going to probably cause you to get Creutzfeldt-Jakob disease. Cases where it is linked, clearly linked, are due to cannibalism. And in this part of the world, we don't practice cannibalism on a regular basis. So rabies, of course, you get from the bites of infected animals, not just dogs, but also squirrels or prairie dogs or raccoons or whatever. The animals don't have to be acting really loopy to be able to transmit rabies. However, if you do see animals that are behaving out of the norm, so for example, raccoons tend to come out during the night. If you see a raccoon out during the day, it's either a really desperate mother foraging for food for her babies, or it might be a raccoon that has rabies. You go up and pet the raccoon and invite it in. Yes, I'm being facetious. No, you don't. You call animal control to deal with the raccoon. I don't know if any of you have messed with a raccoon before, but a raccoon is a mouthful of razor-sharp teeth and a bad attitude. So I would highly recommend you do not mess with a raccoon. And don't have your dog mess with a raccoon either, because your dog will not get the better end of that deal usually. Rabies, as you can see, is a bullet-shaped virus. This is a negative strand RNA virus. It will spread from the bite. It will travel retrograde through sensory neurons. It travels very slowly. It takes months for it to eventually get to the spinal column. When it does, it will infect motor neurons, and the shorter incubation period is if the bite is to the face and the scalp. You see that, or neck, because of proximity to the brain. You see that in kids. Because the movement through the nerves are so slow, you can actually vaccinate the person against rabies. The rabies vaccine used to be very dangerous. Now, the rabies vaccine is genetically engineered. It's very clean, very safe. The old way the rabies vaccine was made, you inject a rabbit with the rabies vaccine, you let it get very sick, You pull the spinal cord out, you homogenize it, filter it, do some isolation bits, and that's your vaccine. Very dangerous. Some people had bad anaphylactic reactions to it and died actually due to it. Not anymore. Not anymore. Rabies vaccine is very safe and much less expensive too. It's much cheaper. So you can vaccinate people. There are also antibodies, monoclonal antibodies against the rabies vaccine that you can give as well that will bind the rabies virus and prevent it from infecting other cells in your body. But if someone is not treated, and kids sometimes, they get bit by dogs and they're embarrassed. They don't want to tell their parents. So if you're doing family medicine, you're doing peds, you see something, even if it looks like a healed dog bite, hey, what happened here? Oh, they're probably playing with the dog. And no, that looks like kind of puncture-ish. There are serological tests for rabies that you can do. There are also molecular tests you can do for rabies. You can see there a handful of people have been known to survive a rabies infection. Don't put your odds on that. The odds are not good. Once rabies gets to the central nervous system, doesn't look good. Really doesn't look good. So both wild and domestic mammals can spread the disease. If you are working in the wild at all, if any of you end up doing wilderness medicine, you will need to be vaccinated against rabies. That is the standard thing. If any of you do any public health work where you deal with wild animals, you'll need to be vaccinated against rabies. That's the standard fare. Skunks, raccoons, foxes, what have you. Out here, it's mainly dogs. If a patient is bitten by a wild animal, you just assume they're going to have rabies and you treat them. You play it safe, essentially. You play it safe, especially now that the vaccine is so safe. You just go ahead, treat them right away. And there's a DNA vaccine that's in development. Last I read of that, it's not actually gone very well. So don't put your hopes on that one. The vaccine that's available now is the surface glycoprotein, and antibodies against the surface glycoprotein neutralize the virus quite well. Grace? Yeah. Yeah. But the first shot gives you pretty good protection. So after that first shot, it is a series, but that's to essentially ensure that you have really robust immunity against it. It is a series, that's correct. But it's after that first shot, it's clear your body will start to clear the virus pretty robustly. Tetanus, of course, you get from puncture wounds. Puncture wounds, usually deep puncture wounds. And the old saw is usually the rusty nail. Doesn't have to be a rusty nail. It could be some garden tools sitting out in the shed that they went outside, they didn't put their shoes on and they stepped on that old rusty rake or something like that. Or they went outside and there was just something sticking up. Oh, darn it, what happened? Thing about puncture wounds, they don't bleed a lot. Because when you puncture it, then you put weight on it and it does that. Oh, it's not bleeding, I just won't worry about it. So tetanus can sneak up on you very quickly. So if someone has a puncture wound, it's caused by a Clostridium. What do you know about Clostridium? They're strict anaerobes. Puncture wounds, they heal over. In there, it's strictly anaerobic. It's a perfect environment for Clostridium. This is a soil organism. So anything contaminated with soil that causes a puncture wound is a prime candidate for tetanus. So, for example, Eric comes in to see you. He's got a towel, a handkerchief wrapped around his hand. Oh yeah, that saw. We left that saw at the work site. I forgot it was there, and I reached up for something and I whacked it. Oh, someone dropped it and it hit me. So first thing that should go through your head is your tetanus booster up to date? If not, let's get it up to date. Secondly, the organism makes a very powerful neurotoxin. This is an exotoxin. This neurotoxin prevents the Meissner cells. Meissner cells are cells that control the alpha motor neurons. They secrete GABA and they keep the alpha motor neurons, which activate muscle contraction, in check. These prevent the Meissner cells from releasing GABA. They prevent the vesicles from fusing with the membrane. Because they prevent the neurons from releasing GABA, your contractions are uncontrolled. The symptoms will start up here in the face. So you start with the inability to relax the face, and you get what's called rhesus sardonicus, sardonic grin, rhesus sardonicus, and then you get lockjaw, trismus. And this could be subtle. People come in, I'm having trouble chewing. I can't chew. So they get this mouth kind of locked in, kind of like this. I'm having trouble chewing, stuff like that. You see stuff like this, first place your head should go is, do you get any puncture wounds? Oh, I stepped on something, but I didn't think anything of it. I didn't see any blood. So let me see that wound. And it might be healed over by that, but once the organism starts to make a lot of this toxin, now you're going to start to get spasms that will start to overtake the trunk. And people will start to flex like this. That's called opisthotonus. And that sort of thing. The spasms can become so powerful that you can actually break bones. And because the diaphragm can't relax, now the patient will die of asphyxiation. It's a horrible death. There is an antitoxin for the exotoxin. Also, the organism is very susceptible to antibiotics. But you have to start treating early. You have to start treating early. The organism is also treated with hyperbaric oxygen because the organism is a strict anaerobe. There's your rhesus sardonicus there. And again, it prevents the muscles from relaxing because it inhibits GABA release by the Meissner's neurons, which are inhibitory neurons of the alpha motor neurons. Babies can get tetanus too if you have a baby that was born under, shall we say, rough circumstances, and the placenta that detaches from the mom gets contaminated with soil or something that was handled, like hands that were contaminated by soil, and then that infected placenta can actually infect the baby. Good vaccination that's available now, and you need to get boosters for this one. So whereas tetanus causes uncontrolled spasms, botulism causes flaccid paralysis. This prevents the release of acetylcholine from motor end plates. Because it prevents release of acetylcholine, now your muscles can't contract. So tetanus, they can't relax. Botulism, they can't contract. Botulism you get from contaminated food. And this is also an anaerobe, Clostridium botulinum. Clostridium botulinum. And your book has these tables that show you the different types of botulism. Infant botulism typically comes because the baby was fed honey, usually before they were a year old. At this time, the baby's normal flora cannot resist the Clostridium botulinum spores in the honey. So Clostridium botulinum becomes a part of the normal flora of the baby. The Clostridium botulinum becomes a part of the normal flora of the baby. It starts to secrete botulinum toxin, and you get so-called floppy baby syndrome. Floppy baby syndrome. So, you future peds nurses out there, you tell the new mothers, never feed your baby honey. Oh, but they really like it. Oh, tough. Feed them something else. You never feed a baby honey. And this is why. Now, their siblings sometimes really like honey, and because their siblings sometimes, especially young girls who are very maternal, and some of you I know are the oldest in your families and you're very maternal. That's a good thing, by the way. Oh, I really like honey, so baby probably likes it too. Oh, put a little on my finger. Oh, they really like it. Here, I'll go. And then the baby all of a sudden has trouble feeding. The baby has poor muscle tone. They thought they were doing the baby some good. And the parent sits there and says, I don't feed the baby honey. So ask questions. Again, don't be accusatory. Don't be accusatory. Please read that article I put on our Blackboard site about how Constance saved my life. It's from the Wall Street Journal. It's a wonderful story. It's very short about how a nurse, a good nurse, can make such a difference in someone's life. A good peds nurse can save a baby's life sometimes by asking the right questions. So again, botulinum toxin, you get this also from foods that were canned and weren't properly sterilized. So canned pears or peaches, sometimes that weren't canned properly, can cause botulism. One of the first signs is usually double vision. There is an antitoxin to this. There's several different isotypes of the toxin. Human disease usually is associated with the A, B, E, and F varieties. There are antitoxins to most of those. Again, this agent is a strict anaerobe. Once you supply the antitoxin, that usually does it, and usually the normal flora of the person usually prevents the organism from gaining a foothold. That's why antibiotics are usually not recommended for cases like that. You neutralize the toxin, usually you're home free. Your net connection is unstable. Which of the following organisms does not cause meningitis? That cause meningitis? It has meningitis in its name. How about this one? That cause meningitis? Haemophilus influenzae? There we go. How about this one? That one? Yes, that causes meningitis in children. How about this one? That causes meningitis? Yes. Yes. How about that one? That causes meningitis? No. Tetanus is a disease of the peripheral nervous system. Peripheral nervous system. And Listeria monocytogenes, that can cause meningitis. So the answer here is D. Your book has these really nice pictures at the end of the chapter. Please look at those. It's a nice review, essentially, of what we talked about. All right. We'll go ahead and take our break. 5 minutes, and then we'll start on chapter 18.
I have a question.
Yeah,
So for lab 53, we don't have some of our labs back to figure out which GI, skin, and throat isolate we had. How do you want us
yeah,
to
I,
like
I
help
have
out?
your labs. They're in
Okay.
my folder. In fact, I brought them to class. I don't know why, but yeah, no, you can hand that lab in later since you'll be back later.
Okay. Perfect. Thank you so
I
much.
could scan them and send them to you. The problem with that is that the scanning is not always very clear. I could try that.
Yeah, that would be awesome.
Let me give that a try. I'll have the secretary scan those since she's better at that than I am. And if that works, then you can do those labs. Do you have those labs with you?
Yeah, we have the lab 53 with me. Us.
Then I will see if scanning works for
Okay,
that.
perfect. Awesome. Thank you.
Yeah. Emma, Reagan, the ladies are talking about staying after talking about the group project. Would you be able to, if I kept the Zoom on, would you be able to do that?
Yeah, I think we can.
Okay. When's your practice today?
I think we're doing something at 10.
Okay, so you can stay, but not indefinitely.
Yeah.
Okay, so 5 or 10 minutes will work. Lisa's gonna say something to you.
Okay, so for the project, we're gonna divide it up. And so since we don't know when you're gonna be back or how much time you have, if you can just send your emails in the group chat so that I can share the Canva brochure to you. And then we assigned both of you editing and photos. If you think you can take on more, you can let us know. But I didn't want to overwhelm both of you. Does that sound good?
That sounds great.
Yes,
Thank
that sounds
you.
awesome. We were just talking about that, and we were a little nervous that doing stuff from here wouldn't be very helpful. So we'll try our hardest to do as much as we can because we really want to help.
Yeah, I didn't want to overwhelm. So if you think you can take on more, you can let us know in the group chat. But if not,
Okay.
both of you will just edit it once everyone's parts are in and then add the photos that are appropriate for it.
Perfect. Yes,
Yeah.
we can do that.
Cool.
Thank you so much.
Yep.
That was painless. Plus, you're in Alabama, you're close to the beach. You're gonna want to take advantage of the beach. Plus, if they win, they're gonna want to celebrate, and they should. No, I can't celebrate. I've got to go do a project. Okay, I'm mean. I'll admit it, but I'm not that mean. I mean, gracious, those are things you tell your grandkids about them when you're in your rocking chair on the porch. Gonna rock something like that? I don't think so.
These are the library view. I just haven't done it. They're all my spreadsheets.
Okay, very good.
I got a question. About that report that we're doing. Do you say we can just write on the back of that lab?
If you can fit it on that back of that, yeah, or you can staple something.
Like this is what it was. This is what it is now that we have. Like it's so like. Like I haven't gone down. So I think we should have someone like focus on that. Yeah, and then like people have to take two vaccines. Just just honestly, here, just do what you want. So we can hopefully bring it to the scene without doing the class. Or you can use 2 or 3 pieces. Yeah, and this is like their.
Okay, it's time to start again. So the beginning of chapter 18 goes into a review of the cardiovascular and lymphatic systems. And I think it's fair that we can skip that because you guys have pretty much seen that. This system is sterile. So it's fair to say that there is no normal flora in this system. So we're going to just skip all that and kind of cut to the chase. So we'll start out with the discussion of malaria. Malaria, of course, comes from mosquitoes. This is a protozoan. It's a member of the protozoan group known as a sporozoa. Oh, okay. That. Should have had Kylie throw it. The sporozoans are not obviously motile. They don't have flagella, they don't have cilia, they don't move by amoeboid motion. They don't have obvious modes of motility. Now, there are different types of malaria depending upon the species, but they all have this very similar pattern. There are members of the genus Plasmodium. The ones that infect humans include Plasmodium vivax, Plasmodium ovale, and Plasmodium falciparum. Most species of Plasmodium that cause malaria infect animals, including birds and mammals. They start out by infecting the liver. That original infection of the liver is very important for the development of the disease. From the liver, so that the insect will inject so-called sporozoites. The disease then goes to the liver, spends a time infecting hepatocytes in the liver. Then you release merozoites to infect red blood cells. When the mosquito takes a blood meal, picks up the merozoites, sexual reproduction occurs in the mosquito. All the reproduction in our bodies is asexual. So sexual reproduction occurs here in the mosquito. And the mosquito bites and injects sporozoites and so on and so forth. The life cycle is complicated. We'll talk more about that and what have you. Now, the symptoms largely arise because the merozoites divide inside the red blood cell, and the infection and destruction of the red blood cells are synchronized to some extent. That burst of the red blood cell causes hemolytic anemia. It also causes a release of lots and lots of cytokines. That's responsible for the fever. So the fever is episodic. The timing of the fever differs between these different species. Cerebral malaria is a complication of typically falciparum malaria. I don't know if any of you knew Nana Yaboa, who was on campus. Yana's by the way, is in medical school now at Liberty in Virginia. Nana actually survived cerebral malaria when he was a child. That's what caused him to want to become a doctor, was that stint in the hospital. In the bed next to him was an 11-year-old kid. Nana was 10. The kid next to him died. He survived. This is very, very severe. Your chances of dying if you get cerebral malaria is quite high. Quite high. All right. So 200 species, 5 infect humans. The big ones are Vivax, Ovale, Falciparum. You also have Knowlesi, and malaria, Plasmodium malariae. Although Plasmodium malariae infects mainly birds. So here's the life cycle we talked about. The species of mosquito is Anopheles. Anopheles mosquitoes, when they bite you, they stand on their head. So if you've ever seen a mosquito, the mosquito just kind of goes like that. That's not Anopheles. Anopheles, they stand on their head as they bite. That's one of the ways you can tell if it's Anopheles. We camped up in Wisconsin one year. The kids were really little then. I came away from that trip with, 36 mosquito bites. My wife had 2. I saw one of the mosquitoes that bit me, and it was standing on its head. I said, that's Anopheles. That's potentially carry malaria, but we don't have malaria in this country, thankfully. And as I smacked it, I said, you're Anopheles, now goodbye. So the Anopheles we're going to essentially start here with the mosquito. So the oocyst ruptures and releases sporozoites. Those infect the salivary glands of the mosquito. The mosquito, when it takes a blood meal, injects salivary secretions that prevents the blood from clotting. That way, as it's sucking up blood, it doesn't clot inside the proboscis. The sporozoites infect the liver, and they mature into so-called schizons. No. schizonts, schizons. The schizons rupture from the hepatocytes, and the liver cells release so-called merozoites. Now it's here we have to note a difference between Plasmodium vivax, Plasmodium ovale, and Plasmodium falciparum. These two species are capable of existing in a liver dormant stage. Why is that significant? It's significant because most of the drugs that treat malaria are going to hit it here. If you have a liver dormant stage, you avoid being killed by most of the drugs that are used to treat malaria. So something to keep in mind. After this initial replication in the liver, the parasites then undergo asexual multiplication in the red blood cells. When the merozoites infect a red blood cell, you see a red blood cell with a ring. You can see these very, very easily on a blood smear. That is still the gold standard for diagnosis of malaria is a blood smear, because that ring-shaped lesion is not caused by anything else that you'll see. So the ring-shaped lesion, absolutely diagnostic. The blood stage parasites are responsible for the clinical manifestations of the disease. When the mosquito takes a blood meal, those particular parasites will mature into micro or male gametocytes and macro or female gametocytes. They will enter the alkaline gut of the mosquito. They pierce the epithelium of the mosquito gut. They then mate in the hemocoel, and they form the oocysts. The oocysts then zygotes are motile. They will then grow in the midgut of the mosquito. The oocysts rupture, they release sporozoites. The sporozoites infect the mosquito salivary glands that then get reinoculated back into you when they bite you. Here are the places where different malarial species are available. We can see South America is mainly Plasmodium vivax. Here in Central America, you have Plasmodium falciparum. Africa, it's largely Plasmodium falciparum, but you have overlap of falciparum and vivax and ovale in some places in Africa. In Asia, it's largely going to be ovale, and then in northern India, it's vivax. In southern India, it's falciparum. Then, as you can see in Indonesia, and over here in Laos, Cambodia, those areas, it is vivax. Then as you get into Indonesia, you get ovale, and then also falciparum bivaccine, so on and so forth. This shows distinct boundaries. Very often those distinct boundaries are blurred and you have overlap in many cases. When you get the merozoites essentially emerging from the red blood cells, Plasmodia metabolize glucose at a very high rate. They can cause hypoglycemia, sometimes very severe hypoglycemia in the patient, even though they're not diabetic. You also get severe anemia. The damaged red cells will cause the spleen usually to enlarge. The protozoa will express different surface antigens, and they're able to evade the host immune system because the host immune system adapts to one surface antigen and then they express different surface antigens. And that's how these things escape the immune system. And they also hide inside red blood cells. Malaria is still a major problem in the world. It's not so much a problem here, but if you do any international medicine, and if you deal with immigrants, which you will in this country, especially with wide open borders like we have right now, you'll have patients with malaria. So here's the belt. We showed you that before. And here are the different types of stages that you get with the different types of malaria. The thing I want you to see is regardless of the species, you get the ring. The ring has different morphologies, but they all have rings. The gametocytes, you'll see that more in the mosquito than you will in the blood, but you see that occasionally. The schizonts that you see in the liver, they all kind of look like a cell with lots of dots, essentially. And these are the merozoites, essentially, in the red blood cells that you see there. Yeah, that's why the blood smear is still the way to go. Antigen tests have been developed. Some of those antigen tests are very good, but still, the blood smear is kind of the gold standard. See the ring-shaped structures here. You don't need fancy stains. A right stain, which is one of the mainstay of blood smears, is the standard. Those easily pick these up. To treat the patient very often, you might have to refer to the patient to a infectious disease specialist. Lots of ways to prevent malaria in endemic areas. You can spray clothing with DEET. You can also use DEET imbued netting for sleeping at night. Sleeping under those can prevent malaria spread. There's a vaccine. It's in phase 3 trials now. The vaccine looks very, very hopeful. It's effective against Plasmodium falciparum, which is the deadliest of these. It's effective in children. Pilot vaccination programs have already started in African countries. So quinine was the mainstay. Most malarial parasites now are resistant to quinine. Artemisinin-based compounds now are the best treatment. And to give you an idea of what's available, this is from the medical letter. That's really so. Can you read that, or is that too small? Okay, I'll just read that to you. A combination drug called Malaria is the kind of the standard treatment for malaria. Malaria is very well tolerated, but it only hits malaria in this merozoite stage, does not hit the liver stage. Now with falciparum malaria, the liver stage ends very quickly, and it passes go, collects $200, goes straight to the RBC stage. So this drug is very effective against falciparum malaria. If you have a dormant form of this with the other two forms, this drug is not going to be effective, but it will clear out the RBC merozoite stage drugs. Same thing with chloroquine. Chloroquine is a good choice, but you see resistance to this drug in various parts of Africa. But it's very good usually in Central America. Doxycycline, pretty standard usually. Doxycycline is good. Don't use it in children. Also, it can increase the risk of yeast infections in reproductive age females. So it's not your first choice as in females, malaromas. In older chaps like me, when I went to Honduras for a mission trip, that's what my doctor gave me. Works just fine. Mefloquine is very effective, but mefloquine again just hits the merozoites. Mefloquine, people who take mefloquine often get very vivid nightmares. So that's something to be aware of. Serious neuropsychiatric disorders have been reported. It's usually the case that people with neuropsychiatric disorders, they're made worse by mefloquine. So that's something to consider if you're giving someone drugs. Consider Malaron instead of mefloquine. Also, you're starting to see mefloquine resistance in parts of Africa. That's something to consider. Primaquine. Primaquine is the only drug that hits the liver stage. So if you have someone with a liver stage, dormant state because of these two species, Plasmodium vivax or ovale, you're going to use primaquine. So this is the one that you're going to use for short-term travel. If you have a patient with glucose 6-phosphate dehydrogenase deficiency, this drug will cause hemolysis, so and jaundice and so on and so forth. So you have to test patients for that if they're at risk of that genetic deficiency. Tefenoquine is a relatively new drug. It's a long-acting analog of primaquine. So this is recommended for areas where you have methylquine or chloroquine resistance. It's effective, and it also is effective against the liver stage stages of those these two, Vivax and Ovale. Any questions about malaria? Incidentally, with falciparum, the cycle of fever and chills is 24 hours. With Vivax, it's 48 hours. With Ovale, it's 72 hours. So either 1 day, 2 days, or 3 days. This brings us to HIV. We won't finish this, but we'll just be able to get started. So this is a retrovirus. Does that mean this is a virus from the 1980s? No, I'm being silly. It's retro because, anyone tell me why? It's an RNA virus. It makes a DNA copy of that RNA. Its DNA copy is then integrated into the genome of the host cell. The cell that it attacks are CD4 positive helper T cells. The CD4 molecule is its receptor for its surface glycoprotein. It infects the T cell goes to the nucleus, the reverse transcriptase. This enzyme reverse transcribes RNA into DNA, and then an enzyme called the integrase integrates it into the host cell genome. And in doing so, because it wipes out the CD4 cells, it completely wipes out the cellular arm of the immune response, creating acquired immune deficiency syndrome. So here are some illnesses that characterize someone infected with HIV. Each of these infections occurs as the CD4 count starts to diminish. Those diminishing CD4 counts make you susceptible to different types of infections. Now, one of the infections that you see in patients with HIV you don't see in anyone else is this organism, Pneumocystis rovecii. This causes Pneumocystis pneumonia. Pneumocystis rovecii is a fungus. It's a unicellular fungus. Only see Pneumocystis pneumonia in people with suppressed immune systems. So if you have a patient, they have this unexplained pneumonia, does not respond to typical antibiotics, you don't get a very good culture. If they have intercourse with other males, because that's the most effective way of transmitting this virus, is males having sex with males, or if they inject intravenous drugs, because dirty needles very effectively can transmit this virus as well, they are in a high-risk group. You should test them for Pneumocystis pneumonia. If they have Pneumocystis pneumonia, you automatically test them for HIV, because they probably have HIV and their immune response is starting to be dulled. Yes, my battery's running low. I can see that. All right, that's all we have time for. So I'm going to keep my Zoom on, but I have to tell you, my battery's running low, so I don't know how long it'll last. So if you guys want to, you already had your conversation? You talked to them already. Okay. So Reagan, Emma, go Cougars. I'm going to end this before my battery dies. Okay.
Thank you so
Perfect.
much.
So
Thank
good
you.
luck, guys. We're pulling
Thank
for you.
you.
Play hard. Bye.